I'm looking where your picture should be, and I don't see you. We're stretching the technology. I'm at Brooks today, and we'll continue with our lecturing. If you have any problems, press your speaker key, your microphone tab, and the individuals in the control rooms at either 1604 or downtown will assist you. As well, at the 1604 campus, everyone met Marco, so he should be there. And at the downtown campus, Nayeli Ayala was there on Wednesday, and I'm assuming she's there today. Again, I have no visual, no auditory, so kind of a technological blind and deaf person. But that's fine. We'll go ahead and proceed. The control rooms, if you don't have my cell phone number, or if there's anybody in the room that has a cell phone and something comes up, you need to get to me. My cell phone number is 367-8953. So I've got it right here in front of me, and so if something comes up, we can always go back to some other technology where you can prompt me to do something a bit differently. What I decided we'll do today is we will switch over to catch up on the syllabus to gametogenesis, Lecture 3, and I will create streaming video to complete Lecture 1 and Lecture 2. Basically, all of these should be review for you straight from Biology 1. So I'm now going to switch to the desktop, and we'll begin the Lecture 3 on gametogenesis. I'm going to make sure my pointer is set at arrow, and we'll move on from here. So we begin with the image of the male reproductive system. This is a mid-sagittal section through the pelvic region of a male. And, of course, a quick review of the male reproductive organs pertinent to our studies, the testes, the epididymis, the vas deferens with the seminal vesicle and the prostate gland, and the lumen of the seminal vesicle being continuous with the ejaculatory duct through the prostate gland, and then to the urethra that has the membranous portion, a prostatic portion of the urethra, membranous, and then the penile urethra. The female reproductive organs pertinent to our discussions today and through the semester are shown here. Of course, the ovary with the various follicles and various forms of development. This looks like a corpus luteum shown here. The fimbria of the fallopian tube with its various portions, the intramural, the isthmus, the ampulla, and the infundibulum, and of course the fimbria are extensions of the infundibulum, or funnel-shaped portion, and these are the structures that will surround the mature follicle as it is pushing out on the surface of the ovary very close to the point of ovulation. The intramural, within the wall, portions of the fallopian tube, of course, open into the uterine cavity. So we had discussed the trek of the conceptus from fertilization, which typically occurs out here in the ampulla, the dilated part of the fallopian tube. So through those initial divisions up until the beginning of the implantation into the endometrial wall, which typically is on the upper posterior portion of the wall of the uterine cavity in this area, is occurring in that first week to 2 weeks. Of course, the uterine cavity is open through the cervical canal and through the external os, cervical os, into the lumen of the vagina. The layers of the uterus are shown here. The smooth, simple squamous epithelial lining on the outside, the mesometrium, the smooth muscle portion, the myometrium, and then the inner portion, the endometrium. So we will follow through with, obviously, discussions of cytology, histology, and organology, consistent with the levels of structural organization, chemical, cellular, tissue, organ systems, and then organismic levels of structural organization. So at present, we are discussing gametogenesis, and of course, spermatogenesis is the term for gamete development in the males, and oogenesis, oogenesis, the way you spell it, and another way of pronouncing it, is the term for gametogenesis in the female. Spermatogenesis, we'll look at figure 2.1 in just a moment, and several others. So it begins at puberty and continues in males until death. So this is obviously, since it's pointed out so particularly, it is something that is distinctive from the female gametogenesis. Spermatogonia is the term for a pool of diploid cells that undergo mitotic divisions. So we will, at the time we discuss the organogenesis and cytogenesis and, of course, histogenesis for the reproductive system, talk about the production of the spermatogonia in the zygote and embryonic stages of development. So these are diploid cells, and of course they will undergo mitotic divisions to produce primary spermatocytes and then leave behind the spermatogonium as a reserve cell, such that more primary spermatocytes can be produced throughout the lifespan of the individual. So this is one of the distinctions in females. The oogonia are no longer present basically by birth. There are only primary oocytes present in the female ovary, the female gamete. So this is really the distinction. There's a developmental difference such that at birth, the male gonad contains spermatogonia only, and the females gonads contain primary oogonia only, and primary oocytes and no oogonia. So the primary spermatocytes then are the daughter cell that is still diploid and will undergo if you guys see that on the screen, hopefully you see it in the control room. This is not good. I'm not appreciating what's popping up on the screen entirely. If you didn't see it, you can contact me later and we'll discuss that. I don't know, I haven't been in here, this facility, for the summer. I don't know how that popped up on the screen. It certainly was not appreciated whatsoever, and my apologies to anyone that was offended by that. The primary spermatocytes are diploid cells, as I said, and they undergo meiosis. Meiosis is in Lecture 2, and as I stated, we'll get a streaming video of that available for your view at a later time. And I hope you were taping that little pop-up thing so we have documented evidence that something weird is going on and there's inappropriate access to our computers here. The secondary spermatocytes are haploid cells, and they undergo meiosis too. So there are the two divisions of meiosis. The primary spermatocytes undergo the meiosis 1, the reduction division, producing identical haploid cells. And these secondary spermatocytes undergo meiosis 2, and they, at the end point of this, will produce spermatids that will, of course, they are haploid cells, the true gametes, and they will undergo final development to become the sperm which are haploid cells, and that final process is called spermiogenesis. So we'll differentiate these stages as we go along of the male gametogenesis. So here we're looking at an illustration titled "The Seminiferous Tubules Are the Site of Spermatogenesis." So spermatogenesis is the term for the entire process. Spermiogenesis is the final phase of maturation of the spermatid to the sperm. So this is very important to make that distinction, because spermatids are incapable of fertilizing the egg, and even the first formed sperm from the final point of spermiogenesis are not motile. With natural fertilization, they would be incapable of projecting themselves through the female reproductive tract. So obviously there's development and then maturation of the sperm that become very important, and they occur at different stages and at different anatomical locations. So we will deal with that as we proceed from the level of the organ down to the level of the cells. So in the left here we see the anatomy of the male gonad, the testes. We see that there's a very thick connective tissue layer around it, the tunica albuginea, with septi, connective tissue septi that's separated into these chambers. Within each of these chambers, of course, is the long coiled seminiferous tubule, and the sperm cells, sperm-producing cells, are within the wall of the seminiferous tubules. Just quickly, before focusing on that, through the rest of the panels from left to right, we will go through the remaining portions of the duct system that we want to identify at this stage. There are several ducts that are in the main portion of the central portion of the testes. We will not identify them at this time. Then there are these small ductili efferenti. I'll use the term, but we don't need to recognize that term at this time. The efferent ducts, if you will, that carry the sperm into the epididymis. The epididymis has a head, a body, and a tail, and the tail leads into the first part of the vas deferens, or the ductus deferens. So the final maturation of the sperm, where they take on motility, is in the tail of the epididymis. So we will see that the spermiogenesis reaches a point where we have a recognizable cell of the sperm, but it is not fully mature until it passes through all of these small tubules into the epididymis and finally into the tail of the epididymis. So it's relative to the size of the sperm itself, it's quite a long distance that it travels before reaching final maturation. So back then to a cross-section through the seminiferous tubule. We zoom in at higher magnification and we see the connective tissue stroma surrounding the seminiferous tubules. The two terms for the components of an organ are the stroma, which is the connective tissue component, and the connective tissue cells that make it up, and the parenchymal cells, those cells that are the functional cells of the organ. So we see some of the parenchymal cells in the connective tissue stroma. These are the interstitial cells of Leydig. They are outside of the seminiferous tubules, among or between them, but not, inside the connective tissue capsule of the seminiferous tubules. Within the seminiferous tubules are the various cell types committed to spermatogenesis. Before proceeding to that, we of course note that the interstitial cells of Leydig are the source of the male sex hormones, testosterone most importantly, which will diffuse, because it is a steroid lipid-soluble molecule, diffuse directly over and bind to the intracellular receptor within the Sertoli cell within the seminiferous tubule. They're not shown prominently here, but there are many capillaries and small blood vessels in the connective tissue stroma, and of course, every organ needs its vascular supply, but any endocrine tissue and organ or gland is going to have a very high, rich capillary bed and high vascularity, such that you can deliver hormones if they're needed for signal, and wash away hormones if they are to be delivered via the blood to other organs and tissues. So now we zoom in on this box within the seminiferous tubule to a higher magnification to look at the histology and cytology. We see first the connective tissue capsule surrounding the seminiferous tubule. So everything is contained within the seminiferous tubule. There are no blood vessels within the seminiferous tubule. They are only present out here in the stroma, as I had stated. There is, in males, a blood-testis barrier, and it is possible for this blood-testis barrier to become compromised, and in some males, they actually produce an immune reaction to their own sperm. So there can be an autoimmune reaction to their own sperm, and it can cause difficulties with fertility. So this connective tissue capsule around the seminiferous tubules is important for that blood-testis barrier. So now we can see the basic boundary just indicated out here of a Sertoli cell. So we can follow the cytoplasmic edges of the Sertoli cell. It is highly infolded and indented in various areas, and throughout it and within it, you could almost think of it as really just forming indentations to wrap around, not really to form an endocytotic or phagocytotic type of a vesicle, but the similar type of thing, just to surround and support the developing sperm cells, or the spermatocytes, undergoing the processes of spermatogenesis. The other term for the Sertoli cells is sustentacular cells. They sustain the sperm-producing cells, and so therefore, surrounding the sperm-producing cells to sustain the activity is important. So the testosterone will be produced by the interstitial cells of Leydig in the stroma, and it diffuses freely across these structures and binds to the androgen receptor. Actually, it's converted to estrogen, and that binds to the estrogen receptor, which targets the nucleus of the Sertoli cells and various genes in there to support that sustentacular function of the Sertoli cell, and thereby testosterone is essential and contributes to the cellular events that we now will basically review. The outermost or deepest cells, this is the periphery. Remember, we have a circular structure, so the deepest cells in the epithelial layer surfaces at the lumen of this tube. The deepest layer is at the periphery of the tube. The deepest layer, most peripheral cells, sperm-producing cells, are the spermatogonia. And we see far over on the right here the spermatogonia, diploid, as we had indicated, 2N, and these eventually will produce, each will divide through mitosis, and we see the proliferation by mitosis. So they proliferate and produce numerous primary spermatocytes. The primary spermatocyte is 2N, so each primary spermatocyte, as we know, will produce 4 of the sperm. And so we'll follow through with that process and double check on my numbers there. So the first meiotic division will produce 2 secondary spermatocytes. The second meiotic division will then double that. So start with 1, produce 2, and then that splits and you end up with 4 spermatids, haploid spermatids, and then they will complete their maturation. You see differentiating spermatids all here at the apex. Remember, the apex of the sustentacular cells is facing the lumen of the seminiferous tubule. So the base of the cell is down here. So the apical membrane is indented and enfolded surrounding the differentiating spermatids. And some of the sperm are at the surface and are breaking away. And then far here on the right, we see the anatomy of the sperm. It has a head at one end and a tail at the other, and a midpiece or body in between. There are characteristic structures in each of these portions. In the head is the nucleus, the haploid nucleus, and the Golgi apparatus has differentiated into this structure called the acrosome. The centriole is at the neck portion, if you want to use that terminology, the junction with the midpiece of the body. The midpiece is loaded with mitochondria, the one site of the synthesis of ATP, much needed, of course, for the cytoskeletal components to produce the powerful swimming action of the sperm. Again, just quickly, the seminiferous tubules in their compartments with the continuity into the epididymis. Here we focus our attention now on the epididymis with the head, a body, and then a tail that becomes continuous with the vas deferens. So the spermatid at the end of spermiogenesis is going to make its way through by bulk flow all of these structures through the output ducts, ductules, tiny ducts, the ductili efferenti, into the head of the epididymis, and then as the sperm, still by bulk flow, are moved through the epididymis into the tail, they mature into motile or fully mobile sperm. And at ejaculation, it is the population of sperm in these areas that are moved forcefully through the vas deferens via the strong contractions, rhythmical contractions of the smooth muscle cells within the wall of the vas deferens. Now we can take a quick look at the development of the male gonads. Not so much that we are studying development of them at this time, but just in the context of spermatogenesis or gametogenesis throughout life. So in the newborn human male, there are just these rather solid cords of cells, the testis cords. The normal mature adult will show these numerous seminiferous tubules that have the highly organized cells in the periphery and the lumen in the center of these tubules. So there is a tremendous amount of development from birth to adulthood to get the final cellular and structural organization, in other words, cytogenesis and histogenesis and organogenesis, to have the mature testes at puberty, at the end of puberty, or during puberty when spermatogenesis is triggered and fertility is established. So this is a high magnification of a single seminiferous tubule, and we see the true density of the cells from the periphery toward the center. And in this, I draw your attention, if you are able to see on your slides or printout once you are looking at it on the monitor as a PowerPoint slide, you should be able to see these dense clumps of structures in the cells at the periphery. And I challenge you to think for a moment, I won't give you too long so we can keep moving, to think of what you're observing. 3, 2, 1, of course you are observing mitotic figures of the spermatogonia. So these represent different stages of mitosis for the spermatogonia to divide and produce the primary spermatocytes. From here on toward the surface, we should see events, I don't even imagine myself capable of picking out any one particular one and saying this is meiosis 1 prophase or meiosis 2 prophase or anything like that. But we know that meiosis is occurring from basically this point toward the lumen. Here we see it illustratively shown. So that at the periphery is the mitosis of the spermatogonia to produce primary spermatocytes. Any cell division event we see closer to the apex of the sustentacular cell is going to be a meiotic event. So this would be some stage of meiosis. This most likely would be mitosis, a dividing spermatogonium. Closer look here, we see this series of events illustratively for the meiosis. The spermatogonium with 46 chromosomes. Here we see the margin of the sustentacular or Sertoli cell all the way to the surface with these deep indentations. All the cytoplasmic organelles are present for this very metabolically active cell, and the nucleus typically is at the basal part of the cell. The apex is near the lumen. So as the cells undergo development, they migrate, undergo the meiotic processes. They migrate from the basal to the apical membrane of the sustentacular cells. So very likely, the sustentacular cells are using their cytoplasmic processes to create and generate this migratory process. Anytime we have a change in the shape or position of a cell or part of a cell, we immediately think of cytoskeleton. So very likely, the sustentacular cells are very rich in free ribosomes producing the cytoskeletal proteins and the cytoskeletal proteins themselves. As we progress from basal to apical throughout meiosis, primary spermatocyte still diploid, 46 chromosomes. Once it divides, the secondary spermatocytes, 23 chromosomes, and after that division, next division, the spermatid, here we see the nucleus is still central, and the cytoplasm surrounds it. The Golgi apparatus is differentiating in directions that appear to be like the acrosome. And then the next stage, the spermatid is beginning to shed, push off and shed its cytoplasm, the bulk of the cytoplasm, and leaving just the nucleus, the pronucleus, as it will become, the haploid nucleus with just the thin rim of cytoplasm and the Golgi, now referred to as the acrosome. So here we begin to see the concept, the philosophy that I have of presenting material within class in the format of exam questions. Don't be surprised if you see these questions on your exam, hint, hint. So the chromosome number of a human sperm cell is, as we saw, it was the haploid, 23, 46 is the diploid. So I try to embed these questions very close to the point of reference for finding the answers to the questions. So the haploid sperm is 23 chromosomes, as we had indicated. So spermatogenesis. So be very careful to look at the final term, spelling of the term. Is it spermiogenesis or spermatogenesis? Spermatogenesis is the all-encompassing. Spermiogenesis is only the final part after meiotic divisions have completed, and the spermatid is produced as it is undergoing differentiation. that is spermiogenesis. So spermatogenesis, we're talking with reference to the entire developmental process. Process releasing mature sperm in the lumen of the seminiferous tubules. That's actually sounding more like spermiogenesis because the focus is on the release of the mature sperm. Involves a kind of cell division limited to gametes. When we think of the first question of producing haploid from diploid, that sounds like a good choice for a correct response. Results in the formation of diploid cells, not ultimately as part of it, so this would not be the best possible answer. We're still left with this second one as the best possible answer of the three so far. Uses mitosis to produce gamete cells. In part, because we start with the spermatogonia, this could be considered true, but it is not complete enough to be the best possible answer, as we see in the third response here. The reason these are not listed as A, B, C, D, or 1, 2, 3, 4 is that they may be rearranged once they are included in the question database, and then again they might be randomly reorganized once they are transferred from the question database into the quiz, any one particular quiz on WebCT. So just a quick review so that we can follow the gametogenesis on the left with this illustration similar to what we had seen previously. Again, our orientation is the basal compartment and the adluminal. So we remind ourselves that when we adhere to something, we stick to it. When we are abducted, we are taken away. So adluminal means that we're close to the lumen. Abluminal, abluminal is far away from it. So the basal compartment is equivalent to the AB or abluminal compartment. The AD, adluminal compartment, is close to the lumen. So events occur in different compartments, subcompartments within the epithelial lining. So the AB luminal subcompartment, the AD luminal subcompartments of the epithelial layer, the surface lining cells called an epithelial layer of the intestines. So the spermatogonia are the deepest cells in the deepest abluminal compartment. They will divide, produce the daughter cell type B spermatogonium. The daughter cell type A remains in the basal lamina as a precursor cell. So it doesn't produce two daughter cells. One spermatogonium produces a cell that goes on to differentiate. I wish they really would clear up the terminology. There's always this gradual change in the terminology. From the old days, we were told that a cell undergoes mitosis, produces two identical daughter cells, and things happen from there. I like to think of it as a reserve cell dividing and producing a cell that will differentiate, and then it stays there as happy as could be, slightly changed because of the semi-conservative process of DNA replication, but that's fine. It's still happy to be who it is and go on with its life. So there is the movement from AB luminal toward AD luminal compartments as it progresses, as we had said. Meiosis 1 occurs, we get the secondary spermatocytes from the primary spermatocyte, which is what the daughter cell type B is determined to become with a little bit of growth. Then there will be, of course, the second meiotic division forming the early spermatids. So from one daughter cell type B, we end up with 4 haploid spermatids that undergo spermiogenesis after meiosis to form the mature sperm. So spermatogenesis includes mitotic division of the spermatogonium, meiotic division of the daughter cell type B spermatogonium, and spermiogenesis for the final maturation or ultimate differentiation of the spermatids to mature sperm. This is figure 2.4 from our text, and again we can focus on spermiogenesis here. We have the haploid spermatid very early, and it undergoes all of these various progressions of cellular change, and basically what we see is all the cytoplasm will be shed, leaving behind basically just the Golgi apparatus that develops into this specialized structure called the acrosome, and it is loaded with degradative enzymes and various structures, molecules that will be important for penetrating the corona radiata and the zona pellucida that surround the secondary oocyte at fertilization. So this is an illustration for purposes of looking at this question. Spermiogenesis involves the formation of 4 haploid cells from a spermatogonium. This is going too far back to be that one. The movement of sperm in the female genital tract, obviously not true. The formation of a functional sperm by the stripping away of superfluous cytoplasm. This is the best possible answer. A little bit more is happening than that, but we're fine with that as the best possible answer because we could not choose this one. So the challenge here is to look at this histological sample and I don't know what that is. So glad you're taping this. I'm just going to click the X on this. I don't know what to do. So here we're seeing connective tissue stroma, connective tissue capsule, epithelial lining of a tubular structure with a lumen. At the surface of this epithelium, we see these long structures here. They might distract you to think that they are possibly the tails of spermatids and that these are sperm that have been freed up. But in fact, these are stereocilia, extremely long microvilli of the seminiferous, of the epididymis, and in the lumen are the clusters of sperm undergoing the final maturation. I'll proceed beyond this. First part basically is review from the illustrations we've looked at. And we note that there are many of these events that we've been describing occurring simultaneously on an ongoing basis in pubescent, post-pubescent, and adult males. So if there are less than 50 million sperm in an ejaculate, this is considered infertile. Sterile is less than 25 million. So the process has to be tremendous. I'm going to leave you to find the answers to these questions. They should be contained within the text shown previously. This slide reminds us that we will see histological samples in this class in the exam. So you'll have to train your eye, establish criteria. The first one here, we're looking at lumen. We're looking at tubular structures with a lumen. We see these long structures in the center. There's a connective tissue stroma, and there is a surface epithelium. It's a thick surface epithelium. The nuclei are all at the base of the cells toward the periphery of this structure in the AB luminal portion. So this satisfies enough criteria to identify this as the epididymis. The process that occurs here is final stages of spermiogenesis as well, final stages, the ultimate stages of spermatogenesis. Both are the same. Again, a histological sample. We have to recognize connective tissue stroma, the edge of an arching structure, very likely some type of a tubule. This would be the AB luminal portion with mitotic events occurring where the spermatogonia are dividing. So here's a higher magnification of all that DNA material changing for mitosis of the spermatogonia to produce the primary spermatocytes, the type B and type A spermatogonia daughter cells. So this is, of course, seminiferous tubule. The process occurring here is spermatogenesis. And so this slide just shows you what you were looking at in the previous one. What is this structure? This, you should be able to recognize this is the apical or adiluminal surface of the epithelium in the seminiferous tubule, and these are the maturing spermatids undergoing spermiogenesis. So the cell type that has the smallest diameter, I'm thinking of cross-sectional diameter of these spermatids that are undergoing the final stages of maturation. So the question here is, what is this structure and what process occurs here? So here we have to recognize this as some type of epithelial lining. I don't see the stereocilia, but we see many sperm out in this area here. So I'll just refer to this as the seminiferous tubule, probably at the junction with the vas deferens. And here is the final stage of maturation where motility is gained by the sperm. So I guess I'm going to have to learn to lecture at a faster pace and stay up on these. I am going to stop here, and because we don't have class on Monday, I'll be very busy at producing streaming videos to get those posted for you as early as possible next week. So my watch is showing 10 to 3, so that's our end point. I appreciate your time and attention. I sure hope you're all there, and I sure hope we get help finding out what's happening on this computer down here so that we don't get pop-ups of the nature, although it's within a class about sex and sexual reproduction, but obviously very inappropriate. And again, my apologies for anyone that might have been offended by that. I know it wasn't horrible, but it wasn't a nice thing to see. So everybody have a good weekend, and I'll see you on next Wednesday. So I will work from here again for the majority of the remaining lectures. In part, we are trying to get our three-way connection working between Brooks, the downtown campus, and 1604. Have a nice day, everyone.